If the last few years have shown us anything, it's the astonishing, sometimes almost unsettling ability of RNA viruses to rapidly evolve,
Yeah.
to adapt.
Right.
Think about SARS-CoV-2, how quickly new variants emerged, keeping everyone, on their toes.
Absolutely.
But what's the real mystery behind this viral shapeshifting? How do these microscopic invaders change so quickly? And maybe more surprisingly, what role do the very cells they infect play in all this?
That's exactly the core question. And today, we're taking a deep dive into something called viral RNA recombination.
Okay.
Now, this is a crucial, but maybe often underappreciated mechanism. It's where viral RNA segments get shuffled, joined together.
Mixing and matching.
Precisely. It's a key driver for new viral variants, for adaptations. And while the viruses themselves, drive the process, host cell factors are surprisingly important. And, well, they've been pretty poorly understood until recently.
And that brings us to our mission for this deep dive with you, our listener.
Hmm.
You provided us with a really fascinating new study. It uses an ingenious and frankly unexpected model system. Tomato bushy stunt virus TBSV in yeast, baker's yeast.
Yeah, sounds strange, but it's powerful.
Their goal, to uncover these hidden host factors and maybe reveal some novel functions of various bacterial and even human viral proteins in the process.
That's right.
So get ready. We're going to unpack some surprising facts and definitely a few aha moments from this really cutting-edge research. So, okay, viral RNA recombination. We hear the term, especially when new variants pop up, but what is it fundamentally? If you're trying to picture this viral shapeshifting, how would you describe it?
Okay, think of it like this. A virus's genetic blueprint, its genome, is often made of RNA.
Right.
Recombination is this basic process where two or more bits from the same RNA molecule, or maybe even from two totally separate viral RNAs, get, well, stitched together.
So not just copying errors, but actual cutting and pasting.
Kind of, yeah. It's like the virus has its own little genetic editing suite. It allows for mutations, sure, but also big changes. Insertions, duplications, deletions, rearranging whole sequences.
Yeah.
It's a genetic playground that lets them evolve incredibly fast.
And why is this genetic playground so important for the virus? What are the, the high stakes here?
Oh, the stakes are huge for viral survival, for spread. Recombination isn't just about creating variety for its own sake.
Right.
It's a powerful survival tool. It can enable viruses to jump to new hosts, expand their territory.
Okay.
And it can even help repair damaged viral RNA genomes. If their RNA gets broken, recombination can sometimes fix it, which massively increases their infectivity.
Wow.
And we see this process in lots of important viruses, including, like you said, SARS-CoV-2.
So how does it actually happen? What's the mechanism?
Well, the main driver is the virus's own copying machine, the RNA-dependent RNA polymerase, or RDRP.
You're the DRP.
Okay. Think of it trying to copy the RNA template. Sometimes it's maybe a bit sloppy, or it hits a roadblock. It might stop copying one template and then jump to a different spot on the same template, or even jump to a completely different RNA molecule and just continue copying from there.
Ah, so it switches tracks mid-copying.
Exactly. That's called template switching. That's how they shuffle their genetic deck so effectively. But, and this is key, what's been overlooked is the role of the host cell itself. It's not just a passive bystander. It plays a big, but until recently, pretty poorly understood role.
Which brings us, squarely to the unexpected hero, or maybe the unexpected lab rat of this study. Tomato bushy stunt virus, TBSV, and baker's yeast, Saccharomyces cerevisiae. Why yeast? That seems like such an odd choice for a plant virus.
It seems odd, but it's actually ingenious. Yeast is a fantastic model system.
Why?
Because its genetics are relatively simple, we understand them well, but its basic cellular machinery, how it handles proteins, membranes, energy, is very similar to ours, to other eukaryotes.
Okay, so it's like a simplified toolkit for studying complex processes.
Exactly. And it allows for these powerful, systematic, genome-wide screens. You can test thousands of genes quickly. In fact, screens like this in yeast have already found over 80 host genes that affect TBSV recombination.
80? Wow.
Yeah. And it's also important to grasp that TBSV, like many other positive strand RNA viruses, that's a huge group, includes things like polio, Zika, dengue, and SARS-CoV-2.
Right.
They all massively remodel the inside of the host cell. They hijack host membranes and organelles to build these specialized structures. We call them viral replication organelles, or VROs.
VROs, like little viral factories?
Perfect analogy. Like pop-up factories built using the host's own materials, its proteins, its lipids.
Yeah.
And it's inside these VROs that the viral replication and, crucially, the recombination happens.
Got it. So the yeast provides a controlled environment to see how building these factories influences recombination.
Precisely. And the researchers here used some really novel strategies to probe those interactions. They had two unique libraries of proteins they introduced into the yeast.
Okay, let's break that down. Part one, they use proteins from the Legionella bacterium. What's the thinking behind bringing bacteria into a virus study?
Well, it was quite clever. Legionella bacteria, the ones that cause Legionnaire's disease, are known to inject proteins, called effector proteins, into the host cells they infect.
Okay.
And these effectors specifically mess with host cell functions. They manipulate pathways to help the bacteria survive and replicate.
Right.
So the researchers hypothesized, what if some of these Legionella effectors, when put into yeast, target the same cellular proteins or pathways that TBSV also needs or manipulates for its replication or recombination?
So the bacterial proteins could act like little spies or probes, revealing the host factors that the virus is exploiting.
Exactly. Probes, that's the perfect word.
And did it work? Did these bacterial spies lead them anywhere interesting?
They absolutely did. It was quite successful. Out of 113 Legionella effectors they screened, 16 of them significantly altered TBSV RNA recombination. We're talking by about 40% or more, up or down.
16. Okay.
And crucially, 7 of those also affected TBSV replication. That suggests they're hitting pathways important for both processes, or closely related ones.
And could they group these hits? Did patterns emerge?
Yes, they could. They grouped them based on the known targets or functions of those Legionella effectors. So they saw effects related to the actin network, the cell's internal skeleton,
Right, cell structure.
also protein synthesis machinery, like elongation factor 1A,
Yeah.
and RAB-GT paces, which are like traffic controllers for vesicles moving inside the cell.
Okay, intracellular transport.
Yeah, and phosphoinositides, those are special lipid molecules on membranes that act like signaling flags.
Lipid signaler, right.
Endosomal trafficking, vesicle movement, and really significantly, a number of hits pointed towards the autophagy pathway.
Autophagy. That's the cell's recycling system, right? We'll
Yeah.
definitely need to come back to that one.
We will. It's a major part of the story.
Can you give us a concrete example? You mentioned one earlier.
Yeah, so for instance, one effector called RAVK, it's known to disrupt actin filaments, that cell skeleton. When they expressed RAVK in the yeast, it increased the amount of TBSV recombinant RNA by about 2.5 times.
Wow. So messing with the cell structure actually boosted recombination.
In that case, yes. It really validated their whole approach using these bacterial proteins, gave them real functional insights into host pathways influencing the virus.
Okay, fascinating approach. What was the second part? You mentioned another library of proteins?
Right. The second approach was maybe even more surprising. They screened proteins from completely unrelated human viruses, specifically SARS-CoV-2 and human metapneumovirus, or HMPV.
Human viruses in yeast affecting a plant virus. What was the idea there?
Similar logic, but maybe broader. The idea was that these proteins from human viruses, even though they evolved in a very different context, might still target common fundamental host factors that TBSV also relies on in the yeast cell.
So they might compete for the same host resources, or maybe block some general antiviral defense.
Exactly. Either competing for essential host factors or maybe inhibiting common antiviral proteins that work against many different viruses.
And what happened? Did these human viral intruders affect the plant virus?
They did. The results were really quite striking. They found 6 SARS-CoV-2 proteins and 2 HMPV proteins that clearly impacted TBSV replication or RNA recombination.
6 from SARS-CoV-2. Which one stood out?
Well, the SARS-CoV-2 nucleocapsid protein, the N protein, was a big one.
The N protein. Okay. Involved in packaging the viral genome, right?
Right. In yeast, expressing the SARS-CoV-2 N protein enhanced TBSV RNA replication by about 2-fold.
Doubled it.
And boosted recombination by an impressive 2 to 6-fold. Specifically for the longer, more complex recombinants, it was about a 4-fold increase.
4-fold from a human virus protein acting on a plant virus.
Yep.
That's wild.
It is. And this N protein was so sort of broadly active that it even stimulated the replication of a completely unrelated insect virus they tested in the same system.
Wow. Any others from SARS-CoV?
Yes. Other proteins like NSP1, NSP3, and NSP5 also enhanced recombination, while a couple, NSP15 and OR7b, actually reduced it. So different effects from different proteins.
Interesting. And the HMPV proteins?
For HMPV, 2 proteins, the phosphoprotein P and the M21 protein, both enhanced TBSV RNA recombination by about 3-fold. And M21 also stimulated replication.
So let's take a step back. What's the big takeaway here? The fact that proteins from human pathogens can mess with a plant virus's replication and recombination in yeast. What does that tell you?
It tells us something really fundamental. The identification of these proteins from such distantly related viruses as modifiers of TBSV strongly suggests they are targeting common shared host factors.
Factors that are conserved across different types of eukaryotic cells.
Exactly. Conserved pathways or proteins that diverse RNA viruses, whether they infect plants, insects, or humans, have evolved to exploit or counteract. It implies that discoveries made in this seemingly obscure yeast plant virus system could have real relevance for understanding human viral diseases.
It points to these fundamental battlegrounds between viruses and cells.
Precisely. These shared host dependencies.
Okay, this is where it gets really interesting, right? You mentioned two major breakthroughs stemming from these screens. Let's tackle the first one. The autophagy connection. You flagged this earlier, it kept popping up.
Yes, autophagy was a recurring theme, hit by effectors from Legionella, and also potentially related to some of the SARS-CoV-2 protein effects.
Remind us again, autophagy is the cell's internal recycling and cleanup crew. For maintaining balance, getting rid of damaged stuff, fighting invaders.
That's the standard role, yes. Cellular housekeeping and defense. But here's the paradox. While autophagy can be antiviral, it can engulf and degrade viral particles.
Right. The cell fights back.
TBSV, like some other viruses, is known to actually hijack the autophagy pathway for its own benefit.
How so?
Primarily, it steals the membranes generated during autophagy. These membranes are rich in specific lipids, phospholipids, that the virus needs to build its replication factories, those VROs we talked about.
Ah, okay. So it uses the recycling system as a part supplier for its factories.
Exactly. But the big question in this study was, okay, we know it helps build the factories, but what's its specific role in the recombination process itself? Is it just about building the VROs, or is there something more direct happening?
Right. Does autophagy actively promote the genetic mixing and matching? What did the experiments show?
This is where they really unraveled it. First, they used those Legionella effectors, RAV-Z and Ligand-9, which are known inhibitors of autophagy.
Okay, blocking the process.
When they blocked autophagy using these effectors, TBSV RNA recombination plummeted. It dropped by about 50% in yeast, and even more dramatically, up to 90% in actual plant cells.
90% reduction, that's huge.
It is. And RAV-Z specifically seemed to work by preventing key autophagy proteins, like one called ATG8, from being recruited to the viral replication sites, the VROs.
Okay, so inhibiting autophagy inhibits recombination. What about the other way around?
Good question. They tested that too. They induced autophagy, basically put the recycling system into higher gear. They used a drug called rapamycin, which inhibits a cellular regulator called mTOR kinase, effectively boosting autophagy. Or even simpler, they just starved the yeast cells of nitrogen, which naturally triggers autophagy.
And then the result?
In both cases, inducing autophagy increased TBSV RNA recombination. With rapamycin, it went up about 70%. With nitrogen starvation, they described the increase as dramatic.
So less autophagy, less recombination. More autophagy, more recombination. It seems tightly linked. What's the mechanism? What's autophagy actually doing to promote recombination?
A key piece of the puzzle came from another protein, ATG2. This protein acts like a delivery truck, transporting bulk phospholipids, those fatty membrane building blocks, to sites where membranes are being formed or expanded, like the VROs.
Okay, the lipid delivery service.
When they overexpressed ATG2, essentially providing more lipid delivery, they also saw enhanced TBSV recombination. This really pointed the finger strongly at the phospholipids themselves.
The membrane lipids.
Yes. But the definitive aha moment came from experiments they did in vitro, outside of cells, using artificial membrane bubbles called giant unilamellar vesicles, or GUVs.
GUVs, right. The microscopic soap bubbles you mentioned, what did those show?
They were crucial. They basically reconstituted parts of the replication process on these artificial membranes, and they showed quite clearly that the specific lipid composition of the membrane mattered immensely for recombination.
Not just any membrane, but the type of membrane.
Exactly. They found that high levels of a specific phospholipid called phosphatidylethanolamine PE, which is known to be enriched in both VRO membranes and the autophagosmal membranes that the virus steals, PE acted as a direct pro-recombination factor. When they made GUVs with low PE content, recombination was massively inhibited. Even the short, kind of aberrant RNA products they sometimes see just disappeared.
Wow. So the virus doesn't just hijack autophagy for any old membranes. It specifically wants those PE-rich membranes because PE itself helps the recombination process happen.
That appears to be the key insight. The co-opted autophagy pathway. delivers the abundant phospholipids, especially PE, needed to build the VROs. And this lipid environment within the VRO directly promotes viral RNA recombination.
That's fascinating. It's about the physical and chemical environment of the factory floor.
Precisely. This was really the first time the critical role of specific membrane lipids, the overall lipid context, and even the protein-to-lipid ratio was shown to regulate viral RNA recombination.
Yeah.
And given that almost all positive strand RNA viruses replicate in these kinds of membrane structures,
Yeah.
this finding likely has incredibly broad relevance. It's probably a fundamental aspect of how these viruses evolve across plants, animals, and humans.
A major piece of the puzzle. Okay, let's switch gears then to the second major breakthrough you mentioned.
Right.
This came from looking at those human viral proteins, specifically the SARS-CoV-2 protein and the HMPV M21 protein. We heard they significantly boosted TBSV replication and recombination in yeast. The big question was, how?
Exactly. How are these proteins from human viruses meddling so effectively with the plant virus? The researchers were meticulous here. They took the SARS-CoV-2 protein and tested its effect in a whole panel of different yeast mutant strains.
Okay, yeast strains lacking specific genes or functions.
Yes. Each mutant was deficient in a different cellular function already known to affect TBSV in some way. They were looking for the mutant where the N protein lost its ability to boost replication and recombination.
Like trying to find the one switch that the N protein flips.
Exactly, and they found it. The key insight the moment the pieces clicked came when the N protein simulatory effect completely vanished, but only in yeast that were missing a specific enzyme called XRN1. That's a 5-beta-3 X-ribonuclease.
XRN1, 5-3 X-ribonuclease. Okay, that's a mouthful. Break that down for us. What is XRN1 and what does finding it mean for how the N protein works?
So XRN1 is essentially one of the cell's main RNA degradation machines. Think of it like an RNA shredder.
A shredder, okay.
It specifically chews up RNA molecules from one end, the 5' end, to the other, the 3' end. It's a key part of the cell's quality control, and importantly, its antiviral defense. It targets and degrades foreign or unwanted RNA, like viral RNA.
Ah, so the cell tries to shred the viral RNA.
Correct. So what this discovery implies is that the SARS-CoV-2 protein, and they found similar results for the HMPV M21 protein, enhances viral replication and recombination primarily by protecting the viral RNAs from being shredded by the host's XRN1 enzyme.
It's like a shield.
Exactly, an RNA protector. They confirmed this further because they found the N protein didn't directly boost the activity of the viral replication enzyme, the RDRP, in test tube experiments. Its main job in this context seems to be shielding the RNA from destruction.
That's a really neat viral counter defense. Did they find any other evidence to back up this RNA protector idea?
They did. They showed the N protein could also protect the RNA of another plant virus, Carnation Italian ring spot tombus virus, from XRN1 activity in yeast.
Okay, so it's not just specific to TBSV.
Right. And it even boosted the replication of a native yeast virus, the LADS RNA virus, which is known to be a natural target of XRN1.
So it generally protects RNA that XRN1 would normally degrade.
Yes. And the HMPV M21 protein seemed to work in a very similar way, actively blocking XRN1's antiviral function. This really uncovered a novel function for these human viral proteins. They've evolved this strategy to counteract XRN1.
Which tags XRN1 itself as a really important host defense factor, a restriction factor against RNA viruses.
Absolutely. A key player in the cell's intrinsic immunity that these viruses have found a way to neutralize. So when you step back, this deep dive really gives you a powerful glimpse into that intricate arms race, the constant back and forth between viruses and their hosts.
Yeah.
And understanding these specific cellular weak points, these vulnerabilities, and the clever counter strategies viruses use is just, well, crucial.
Yeah.
Especially with new viral threats always emerging.
And it really highlights the power of their approach, using TBSV and yeast as this kind of cellular system sensor, as they put it.
It's a great phrase.
It seems like this method could be used much more broadly in the future, right? To quickly figure out what various bacterial toxins or even proteins from other viruses are actually doing inside a cell. It's like a sensitive functional assay.
Absolutely. It has huge potential for discovery.
Yeah.
It's also worth remembering from this study and others that the picture is complex. Some host factors affect both replication and recombination, maybe because the processes are linked.
Okay.
But others seem to uniquely influence just one process. So teasing apart these specific roles is an ongoing effort. It's not always straightforward.
And we should, of course, mention the usual caveat. Findings in yeast, while providing fundamental insights into basic mechanisms, might not translate exactly to human cells. There will be differences.
That's true. There are always specifics to the host system.
Yeah.
However, the core processes we're talking about, autophagy, RNA degradation pathways like XRN1, these are highly conserved across eukaryotes.
Meaning the fundamental mechanisms they uncovered here are very likely relevant for a whole range of viruses affecting plants, animals, and yes, humans.
Highly likely, yes. These discoveries have profound relevance well beyond yeast and plant viruses.
Okay, let's try to bring this all together for you, the listener. Let's unpack the big takeaways. We've really seen two major breakthroughs in understanding viral RNA recombination.
Yeah.
First, that really surprising role of the autophagy pathway. It's not just passively supplying membranes for viral factories.
Right.
It's actively promoting recombination by enriching those factories with specific lipids, especially that phosphatidylpolylitholamine, PE. It's like providing high-octane fuel for viral evolution.
It's a fantastic finding. And what's equally fascinating is the second breakthrough. Learning how these proteins from human viruses, the SARS-CoV-2 N protein and the HMPV M21 protein, give viruses a boost.
Yeah, the RNA protectors.
Exactly, acting as RNA protectors, shielding the viral genetic material from the host's own antiviral shredder, the XRN1 exoribonucleus. These insights really pinpoint crucial host factors and reveal these, well, quite sophisticated viral strategies.
So this naturally leads to a final, maybe provocative thought for you to consider. What other essential cellular pathways are viruses secretly hijacking or manipulating in ways we haven't even discovered yet? And what does uncovering those secrets mean for our ability to fight future pandemics, future disease outbreaks, and maybe even for our basic understanding of how cells and life itself work?
It's a crucial question. We really encourage you to think about this constant evolving dance between viruses and the cells they infect. It's happening all the time, inside us, around us. And it's innovative research like the study we discussed today that helps us begin to understand, predict, and maybe, just maybe, intervene more effectively in this fundamental biological conflict.